No, ma'am.
Lack operon was clear to all of you, right?
Yes, ma'am.
Oh. Just 1 second, students. Thank you, Ikra. Thank you. Thank you very much. All right. Okay, so let's come on to this particular thing. Now we were doing DNA fingerprinting. Now, so see, DNA fingerprinting. Have you people ever heard about DNA fingerprinting? None of you have. Okay, see. Now, like while we were doing the human genome project, you people got to know the fact that every single individual and their genome, they are almost the same for all the individuals, but we only differ from each other from that 0.1%, right? So because of that 0.1%, today we're going to see how it is, like how it functions and everything about it, okay? So as stated in the preceding section, meaning while we were talking about the human genome project, 99.9% of base sequence among humans is the same. All of us, we have the same base sequence, but only that 0.1%, that is what makes us different from each one. Now, assuming human genome as 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? You can do the math. Now, it is these differences in the sequence of DNA which make every individual unique in their phenotypic appearance. What is phenotypic appearance, students?
Physical appearance.
Very good. Physical appearance. Everyone knows what phenotype is, isn't it? Okay, very good. Now, if one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine, obviously, because once the human genome was sequenced, it took how many years? It was started in 1990, right? And then it got completed in 2003. So again, it is going to take a lot of time. And when a person wants to do a quick analysis, right? So then came the DNA fingerprinting. Now, DNA fingerprinting, it involves identifying differences in some specific regions in the DNA sequence called as the repetitive DNA. Because in these sequences, a small stretch of DNA is repeated many times. Now, these repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient centrifugation. You people remember while we were doing that transformant experiment, when the genetic material, when it was grown on heavier nitrogen isotope, it got to the heavier side. You remember after centrifugation, right? So when this was done, that density centrifugation was done. So the bulk genomic DNA had different peaks and the repetitive DNA had different peaks. Now these repetitive DNA are these, you can say, prime importance for us. Now the bulk DNA, it forms a major peak in that centrifugation, and the other small peaks are referred to as the satellite DNA. Now, depending upon the base composition, meaning what is the richness, whether it is adenine is to thymine rich or whether it is guanine is to cytosine rich, then the length of segment and the number of repetitive units, the satellite DNA is classified into many categories such as microsatellite, mini satellite, depending upon all of these factors. So you should know that, okay, which are the factors by which we can differentiate amongst the different kinds of satellite DNA. Now, these sequences normally do not code for any protein, but they form a large portion of human genome. Now, these sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Which sequences? Meaning these repetitive DNA sequences, what they do? They do not code for any particular protein, but they form a major part or a very large portion of the human genome. That is why they are. And also, what is polymorphism, students? If you see the word polymorphism, what does it indicate to you or denotes? Break the word and tell me. Mariam? Good to see you back.
Poly is many. And morphism.
Morph. Have you people seen Power Rangers? Are you
Like
from that?
changing?
Changing. Yes, morphing, changing into different types, right? Power Rangers used to morph into Rangers, right? Everyone who has seen Power Rangers know. So same way, these DNA, these repetitive DNA, they do the, you can say they have high degree of polymorphism, meaning they can change the way they are. Okay. Now, since DNA from every tissue, such as the blood, the hair follicle, skin, bone, saliva, sperm, anything from any of these tissues from an individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphisms are inheritable from parents to children. So that is why in the case of like disputes and when people want to have paternity tests done. So this polymorphism, since it is inherited, meaning if the father has it, meaning the DNA or the repetitive DNA which was present in the father, now the child will have. So if the child has the same repetitive DNA as the father, then that means that he is his son or she is whatever. But this is the basis of paternity testing. Okay, because it first of all, the DNA, these DNA that are prime importance of DNA fingerprinting. First of all, they are not coding, but they form a bulk of the genome. Then, secondly, they show a high degree of polymorphism. And apart from all of that, they can be inherited from the parents to the offspring. So that is why. Now, as polymorphism in DNA sequence is the basis of genetic mapping. Remember this, students. Polymorphism is the basis of genetic mapping of human genome as well as of DNA fingerprinting. So if anyone would ask you what is the basis, like what is like, whenever we do any kind of experiment or do any kind of thing related to, and if we want to test something, so there is a basis of our testing, right? If we want to see that basic proteins are present, so we should know the characteristics of basic protein. So that is why same here, polymorphism is the basis of DNA fingerprinting. Now, it is essential to understand that it is essential that we understand that DNA polymorphism means in simple terms. See, polymorphism is variation at genetic level. It arises due to mutation. So at every level, it would show different kind of forms. Poly means many and forms. Now, new mutations may arise in an individual either in somatic cells or in the germ cells. We know that. What are somatic cells? What are somatic cells, students?
They are the lining present in the
They are the lining.
There are many
There are
terms.
these cells from where all the organs arise. Or reproductive cells.
Then what are germ cells? Germ cells are reproductive cells. Somatic cells are body cells. So mutation can arise in somatic cell also. They can arise in germ cell also. Now, if a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, then it can spread to the other members. Meaning, okay, let's just say that during the reproduction process, there was some kind of mutation in the gamete, but that mutation was not deadly or negative mutation. So since it was not, so it got inherited from the parent to the offspring. So it can spread to the population just like that through the method of sexual reproduction. Clear? Now, okay. Now, allelic sequence variation has traditionally been described as DNA polymorphism if more than one variant, that is allele, at a locus occurs in human population, meaning that is how it spreads basically. Okay, now with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, then it is referred to as DNA polymorphism. When it is being inherited from the parent to the offspring and it is happening, then we'll call it as DNA polymorphism. Now, the probability of such variation to be observed in non-coding DNA sequence would be higher as mutation in these sequences may not have any immediate impact, meaning they are basically here trying to tell you that only those mutations are going to be inherited which are not detrimental. You people understand the meaning of detrimental? Meaning which is not harmful to the human being. Now, these mutations, they keep on accumulating generation after generation, and they form one of the basis of variability or polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide change to a very large scale changes, because you people have studied mutation. So now you know what are the various kinds of mutation, right? Point mutation is there, then frame shift mutation is there, isn't it? So all of these mutations, you can count them in the different types of polymorphism. Now for evolution and speciation, such polymorphisms play very important role, and you will study these in higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. Very important to know the person's name who developed the DNA fingerprinting technique. Now he used a satellite DNA as probe that shows very high degree of polymorphism. It was known as VNTR, that is variable number of tandem repeats. Many a time the full form of this is also asked. Okay. Now the technique as used earlier involved the southern blot hybridization using radio labeled VNTR as a probe. Now see what happens is that whenever we are trying to work with DNA, okay, or any molecule, so since we take a lot of it for testing, so what we do is that we label it with a radioactive molecule. Now, if there is any substance, let's just say DNA has so many substance, right? Nucleotide is there and everything. So if we take the nucleotide and imagine that you're putting a tag on it of radioactivity, so it will show, right? Imagine you're taking somebody, okay, and you're doing the experiment in pitch black or dark. Now you're putting, like, have you seen indicator lights on the road? The fluorescent lights. Now, if you wrap that person in that light, will you be able to identify that person? Of course, that person is going to shine. Yes, you people did not understand what I said. It is a very weird example, but okay. For example, you're testing something in a population. Okay, you have 5 subjects in the population. Subjects means people. Now you give them a particular red colored flag or green colored or blue colored flag, each of them to hold one colored flag. And the rest of the population does not have any flag. So you will know which one you are testing. So that flagging is what is known as radio labeled VNTR. We call them as probe. Anything that is a probe, it means that it has been radioactively labeled, meaning it has been mixed with some radioactive substance. Now let us see what happens while we do the process of DNA fingerprinting. But before that, see, they have written southern blotting. Anyone knows what is southern blotting? See, to identify DNA, to identify RNA, and to identify protein, blotting techniques are done. Blotting, you people understand what is blotting in general? Girls might understand blotting. Has no, none of the girls also understand. Has any one of you used those Sheets that absorb oil from your face, blotting sheets. Yeah, you have also used it, right? So that is blotting. What do you do when you do blotting? You place it on something, right? And then it absorbs the oil, or it gets transferred to that sheet of paper, right? So we are going like in during the southern, northern, and western, three types of blotting techniques are there. Now don't imagine it like blotting paper. It's not blotting paper. Blotting refers to transferring. Okay, so during the like analysis of DNA or analysis of RNA or protein, we do blotting techniques. Okay, now this you can write down in your notebook somewhere in a box. Now blotting techniques are of three types. We have northern blotting, we have southern blotting, and we also have western blotting. Okay, and each like one of them is for DNA, one of them is for RNA, and one of them is for protein. Now I'll give you such a nice trick that you will always remember and praise me forever. I'll tell you what the trick is. Whenever, like, this thing will also come when you go for bachelor's. Any graduation that you do, if it is in the field of science, you will get this. So if someone, like, how would you know that northern is for some kind of other blot, which one is the blotting for which? So for that you will do this. You will write snow. Okay, you'll write snow, and then you'll write drop. You'll write snowdrop like this, and then you'll cross out the O's. Now southern blotting is for DNA, northern blotting is for RNA, and western blotting is for proteins. Clear? This helps me even now. Even now also, this is my favorite trick ever. So you'll write snow and drop. Clear to you all? This trick is nice, no? It's easy peasy. All right. I don't know what happens to my pen. It stops working mid class. So you'll write. So you'll write SNOWDROP. You'll cross out the zeros or O's, and then you know that southern blotting is for DNA, northern blotting is for RNA, and western blotting is for proteins. Clear to you all? All right.
Yes.
Okay. Now coming on to these steps. Now let us see what are the steps in the technique of DNA fingerprinting. The first thing is isolation of DNA. Now, DNA can be isolated from a variety of sources. How many of you have seen CID? The Indian television show CID. None of you. You have seen it, like you know what CID is about, right? Don't be ashamed. I know it's very cringe, but tell me if you have seen it. None of you have seen CID. You have, yeah?
Yes,
It's not
ma'am.
a shame to accept
No,
that.
ma'am. It's not.
What about others? No one has seen CID. How will I give you a reference then? Okay, anyway, you people have seen detective shows, any kind of detective shows? Okay.
Yes, ma'am.
Okay. Any detective shows that is associated with crime. So have you seen that people, they wear gloves and they go to the places, and with a cotton swab they take things and they put it into a file, and then they send it for testing. What are they doing? They are doing DNA fingerprinting only, right? For example, if you want to know whether who did the murder, then maybe in the crime scene, let's just say for the handles, they swab the handles also, right? Anywhere that traces of DNA can be found. If they find a piece of hair, that is also very important, because anything can be, you can say, a DNA source, right? Just as we saw blood, saliva, sperm, hair, anything. So first, DNA gets isolated. Isolation of DNA takes place. We'll discuss more about this again when we will talk about biotechnology. Then what happens? Digestion of DNA fragments by restriction endonuclease. Then we break the DNA fragments. That is what digestion means. Then the broken DNA fragments, they are separated. So we do electrophoresis to separate them. Right now, you don't have to worry about each of them. You'll study these in detail. Now we do the blotting technique. Now, since we are trying to identify DNA, which type of blotting will we do? Northern, southern, or western?
Southern.
Southern
Southern,
blotting.
ma'am.
Very good. Exactly. Now, so we'll do the blotting, and then hybridization is done when we label the VNTR probe. And lastly, what happens? The hybridized DNA goes for autoradiography. Now, when all of these techniques are done, bands of DNA are produced like this. Now, now in case of like paternal, like this maternity dispute or something of that sort, then the DNA testing is done. So it is matched with the already known set of DNA. And if it comes out clearly like the same, then a person knows that, okay, this is where the person belongs. Now, the VNTR belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. Copy number means how fast the DNA can replicate or multiply. Okay. Now the number of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Now see students, as far as this DNA fingerprinting is concerned, you don't have to sweat too much on each of these things. Okay. No in-depth question will be asked from DNA fingerprinting as far as I know. Just read it so that you have the knowledge to answer these questions. But the in-depth or to ask the steps, these things will not be asked. Now, the autoradiography, when it is done in an autoradiogram, it gives many bands of different sizes. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population, except in the case of monozygotic twins, meaning they are identical twins. So their DNA will not differ a lot. Can someone tell me why? Why in the case of monozygotic, meaning identical twins, why does the DNA not differ from each other? I need a very good reason, a very solid one, a very scientific one. Don't tell me, ma'am, they are identical, that's why. No. Why? You can give it a try.
Because
Anyway, say.
because it's the same sperm. It's from the same sperm they're made.
They are from the same sperm. Okay, more. Okay, you're
Oh God.
close. Yes, yeah. Are you saying something? See, students, what happens in the case of monozygotic twins? You're very right, Mariam. You're very close to it. What happens when an egg and and a sperm fuses? A zygote is formed, right? Now, this zygote, before starting to differentiate into an embryo, it divides or splits into two. So imagine one zygote is there, it got split into two, and then each of the splits or the halves of the zygote will differentiate. So they are, like, you can say they are coming from one egg and one sperm only, right? They have the common sperm, they have the common egg. That is why the identical twins have their DNA same. Okay. Now the sensitivity of the technique has been increased by the use of polymerase chain reaction, PCR. Again, you will study about this in biotech. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. Why? Because, because of technology and its development, if a person, like even if a person has a tiny bit, like even from one cell, imagine cell, we have an organ system. Imagine one cell. So DNA from that one cell is also enough. Why? Because we'll take that one cell, we'll do PCR. And when we'll do PCR, 1 billion copies of that DNA can be made. 1 billion. That is why it's very easy to do so. Okay, even if we have a tiny trace, we can do so. Now, in addition to application in forensic science, it has much wider application, such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. Okay, so what are the three basic uses of DNA fingerprinting? We have the first one, the first and the most important one, forensic science, to to find out any kind of dispute, like paternity dispute, then rape cases, like majority of the things in terms of forensic science, any crime related stuff can be identified from. Apart from that, it is also used in determining the population and determining the genetic diversities. So with that, we come to the end of the chapter. This chapter is done. Now, as far as the lack operon notes goes, I'll give you the notes, like I'll send the PDF to everyone. Okay, I'll not waste time to recite it and you people will note down. This much is clear to you all? Any doubts anywhere?
Yes, ma'am.
No doubts, right?
No, ma'am.
Okay, I'm very glad. All right. So, like, what is the current chapter going on in your school? Each one of you, tell me. What all chapters are you people on?
I have holidays.
Oh, all of you. Yeah, summer vacations must be going on, isn't it?
Yeah, yeah.
For all of you? Okay. Ikra and Amna, for you also?
Ma'am, I'm doing it online, so
Yours is online schooling.
yes.
All right. Okay, yeah, you have your school going on, or is your holidays are your holidays going on? Holidays? Oh, his network connection is bad today. Okay, so we'll start a new chapter now. Okay, just give me 1 second. I'll start a new chapter.
Ma'am,
Yes.
can I pray and come back after that?
Okay. Okay, students,
Ma'am,
can you see my screen? Yes, yeah.
yes, ma'am. You were asking me something before I got kicked out.
Yeah, I was asking whether your holidays are going on, whether your school is open.
Oh no, my holidays just started.
Okay. And what is the last chapter that was being done while your school was closing?
We just started evolution, ma'am.
Oh, evolution.
We can also do this chapter also, ma'am, because in my half-yearly exam, this chapter is also counts, human health and diseases.
Yeah, we'll start this chapter only, human health and diseases. Okay, students. Are you people preparing for your test tomorrow? Not tomorrow, Tuesday?
Yes, ma'am. I'm not going to write it on Tuesday. I'm going to write it.
Hmm?
I'm going to write it on Saturday.
It's okay whenever you want to, but prepare for it, okay? Don't be unprepared and give these.
Ma'am, is it okay if I off my camera? My internet is
Hmm.
not working again today. Thank you.
All right, just 1 second, students. Okay, till then, students, you write something about the lac operon. Okay. You write down about the operon concept. Okay. You write down about that. All of you switch on your videos. Amna, Ikra, Siddika, switch on your videos. Okay, Amna. All right. Okay, students. Now you write down. Okay. First, you write down who discovered or who gave the lac operon system. So you write down their names. That it was Jacob and Monod, but you write down it was who Jacob. So Francis Jacob and Jackie's Monod. Okay, write this down. Francis Jacob and Jackie's Monod. They gave the operon model in bacteria. They gave the operon model in bacteria. Okay, they gave the operon model in bacteria. Then,
No, ma'am, 1 minute.
okay, they gave the operon model in bacteria.
Then
Then you write down operon is a coordinated group of genes. Operon is a coordinated group of genes such as operon is a coordinated group of genes such as structural gene, comma, operator gene, comma, promoter gene, comma, regulator gene,
Ma'am, what was after operator gene?
promoter gene, then regulator gene, which function together.
ma'am,
Which
after promoter gene,
after promoter gene you have regulator gene, which functions together and regulate a metabolic pathway, which function together and regulate a metabolic pathway as a unit. Done. Then
Yes,
you
ma'am.
write down example, lac operon. In the bracket you write down lactose, that the lac is for lactose. Okay, lac operon. In the bracket you write down lactose. Then after that you write down TRP operon, that is tryptophan operon. What is tryptophan, students? Anyone remembers what tryptophan is? Is it a fan?
No.
Then what is it? It is an amino acid, okay? So for after lac operon, you write down TRP operon, that is tryptophan operon, comma, ARA operon, that is arabinose operon. Then you write down his operon, HIS, his operon, that is histidine operon. Histidine operon. Then you write down val operon. Like you should know the full form of each of them or what they stand for. So this is valley. Okay, so we have tryptophan operon, arabinose operon, his operon, and val. Then you write down etc. There are many more. Then
Yes, ma'am.
everyone's done. All right. Now we're going to talk about, like, we wrote so many different kinds of genes, right? We wrote about regulator gene, operator gene, and everything. So now we're going to talk about each of those genes. So first you write down regulator gene. Write down about one regulator gene. Put a dash, then you write down about it. Tell me once you're done. I'll say that also.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Okay. Now you write down, it synthesizes a biochemical. It synthesizes a biochemical. It synthesizes a biochemical or regulator protein or regulator protein or regulator protein which can act positively, which can act positively as activator, which can act positively as activator and negatively as repressor. Negatively as repressor, full stop. Done? Shall I repeat?
Yes, ma'am. No, ma'am.
Everyone is done, right? No one needs
Yes,
repeating?
ma'am.
Okay. Then you write down the next line. It controls the activity of operator gene. It controls the activity of operator gene. Full stop. Done?
Yes, ma'am.
Yes, ma'am.
Okay. Then the second one you write down operator gene. Talk about operator gene. You write down it is a gene which receives the product of regulator gene. Full stop. It is a gene which receives the product of regulator gene. Full stop. It allows the functioning of the operon. It allows the functioning of the operon when it is not covered by the when it is not covered by the biochemical produced by the regulator gene. When it is not covered by the biochemical produced by regulator gene. Full stop. Shall I repeat this whole thing again?
Ma'am, by the biochemical.
Produced by regulator gene. By the biochemical produced by regulator gene.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Okay. Then you write down third about promoter gene. Promoter gene. It provides attachment site. It provides attachment site for RNA polymerase. It provides attachment site for RNA polymerase. Full stop. Done?
Yes, ma'am.
Okay. Then you write down fourth, structural gene. Structural gene. In that you write down it transcribes mRNA. It transcribes mRNA for polypeptide synthesis. For polypeptide synthesis.
It transcribes.
mRNA, messenger RNA, mRNA for polypeptide synthesis. And when you write polypeptide synthesis, in the bracket you write down protein, because protein is the polypeptide that we are talking about. So it transcribes mRNA for polypeptide synthesis.
Done, ma'am.
Everyone's done?
1 minute.
Okay. Done?
Yes,
Yes,
ma'am.
ma'am. Done.
Okay, very good. Now your heading should be the lac operon. The lac operon. Now in that you write down another the lac operon consists of one regulatory gene, or inhibitor gene. The lac operon consists of one regulatory gene or inhibitor gene. And when you write that in the bracket, you write down I. So this is the regulator gene also, and it is the inhibitor gene as well. So the lac operon, it consists of the regulatory gene or the inhibitory gene, comma, one promoter gene. It has one promoter gene, comma, one operator gene, and three structural genes.
Ma'am, after promoter gene?
I'll repeat it again. The lac operon consists of one regulatory gene or inhibitor gene, comma, one promoter gene, comma, one operator gene, and three structural genes. And three structural genes. Done. And what are the
Yes, ma'am.
structural genes?
Yes.
Does anyone remember the names of the structural genes?
Chain.
Tell me. Z, Y, A. In the bracket, you write down Z, Y, A. Who remembers what they synthesize? None of you remember that. Okay. This much is done. Put a full stop. Now, the next line is a polycistronic structural gene. A polycistronic structural gene is regulated by a polycistronic structural gene is regulated by a common promoter and regulatory gene. A common promoter and regulatory gene. Done?
Yes, ma'am.
Yes, ma'am.
Okay. Now change the paragraph and you start writing that in E. coli, comma, breakdown of, in E. coli, the breakdown of lactose requires three enzymes. The breakdown of lactose, it requires three enzymes. Full stop. The breakdown of lactose requires three enzymes. Now, these enzymes are synthesized together. These enzymes, they are synthesized together in a coordinated manner. These enzymes are synthesized together in a coordinated manner by functional unit of DNA. By functional unit of DNA. Then you write down that is lac operon. This is the
Ma'am,
structure. Yes.
can you repeat after synthesized together?
These enzymes are synthesized together in a coordinated manner by functional unit of DNA. These are synthesized together in a coordinated manner by functional unit of DNA, that is lac operon. This lac operon. Full stop. Done, students?
1 minute, ma'am.
No, ma'am.
Done, ma'am.
Okay. Okay. Okay, then keep it till that only. Let us start this chapter now. Okay. Done? Everyone's done. I'll send you the rest of the pictures that are required for this particular notes that you may be needing. But till here, all of you are done.
Yes, ma'am.
Any doubts up until now?
No, ma'am.
All right. Okay. Okay. So now today we're going to start a new chapter. Now see, I know that in terms of a series or in terms of a sequence, after molecular basis comes evolution. But if you people hated molecular basis that much, I don't know what you will do to evolution. So I'll leave evolution for now. I'll start with human health. All of you will be interested in this. Since this is all about diseases, you people can relate, you people can understand it better. We'll see a lot of pictures also in this particular chapter. So it will be very interesting to all of you. Okay. So we'll do this particular chapter, that is human health and disease. We only have, I think, half an hour left for the class to be over. Okay. Now, talking about human health and disease, the first thing that we must remember, that who is the father of medicine? So Hippocrates, he is the father of medicine. Okay, Hippocrates. Now, after that, there comes who discovered the blood circulation. So it was done by William Harvey. William Harvey discovered the blood circulation. Now, when we talk about human health and diseases, so first thing that we need to know is that what is health? Now, health is a state of complete, meaning physical, mental, and social well-being. When a person is physically fit, mentally fit, and also socially fit, then only a person can be called as a healthy person. If a person is physically fit and can socialize with others also very nicely, but has some mental health issues, then the person will not be considered as healthy. So a person who is healthy, that person has a state of complete physical, mental, and social well-being. Then only you can call that person healthy. Now, health can be affected by variety of things, out of which three are the most important ones that come into our mind. The first is obviously any kind of genetic disorder, meaning the disorder that the human has been born with. Those are the genetic disorders. After that, there is infections, right? Now, the person has been born normal without any disease or anything, but the person got infected with some virus, bacteria, all of us have had that. Now, that is the second thing, or the lifestyle. The lifestyle that we have, whether we have an active lifestyle or whether we have a sedentary lifestyle, all of this affects the health. So these are the three major, you can say, groups of area or broad regions which by which the health can be affected. Now, what are the things that you must do to improve or maintain the good health that you have? Must have a balanced diet. Now, a balanced diet is a diet that consists of all the nutrients in a very appropriate amount, such as it must have carbohydrates, proteins, fats, vitamins, minerals, roughage, and also water. Then personal hygiene. Many a times, many people have fungal disease and all because of the lack of personal hygiene. So personal hygiene is very important. Cleanliness is very important. And also regular exercise. Be it for any motive or just like that. Regular exercise again, very important to maintain the good health. So these are the three basic things. So health part is clear to you all?
Yes, ma'am.
Yes, ma'am.
Okay. Now coming on to diseases. Now, when the functioning of one or more organs or the system of the body, it is adversely affected, then we can say that we have a disease. Now, the disease does not necessarily mean the whole entire body is infected. No, The disease, like for example, there are so many diseases that are respiratory disorders, right? So they only affect the respiratory system. Will they affect the digestive system? No. Will they affect the excretory system? No. So the disease, it can affect one organ or more organs or a whole system also. So that is what disease is. Then we can say that we have a disease. Now, disease, they can be of two types: infectious disease and non-infectious disease. Now, infectious disease can also be called as communicable disease, meaning it can spread from one infected person to a healthy person. And when we talk about non-infectious disease, means they are non-communicable. They cannot spread from one infected person to a healthy person. The best example for infectious disease is common cold. Like there has been so many cases, like you can say almost all of us once in our lifetime have been very healthy, without any disease. We went to the school, any classmate had a cold, then we also caught a cold, right? This has happened to every single person. So that is an infectious disease. Another very recent example is coronavirus, right? COVID. If one person had it, everyone had. Now, talking about the non-infectious disease, meaning they cannot spread from one person to another, and that is cancer. If one person has cancer, no one else can get cancer from that person. So these are the classes of diseases that we have. Now, when we are talking about disease, so we must talk about the elephant in the room, the pathogen that caused the disease, isn't it? So pathogens are those disease-causing organisms. Now, they come in variety of groups. They can be bacteria, they can be viruses, they can be fungi, they can be protozoans, and they can be helminths. Now, before I move forward, I want one scientific name from each of these groups. Tell me one scientific name of any bacteria, any bacteria. Any bacteria. You can tell me the name of any bacteria that you want, that you like. The question is to everyone though. All those that have their video switch on, I'm not saying anything because it's almost ending, like time is over, but tell me the example of any bacteria. Now, this is the easiest question that I could ask to people. Easiest. Tell me, Ikra, what about you? Any bacteria example?
Streptococcus.
Streptococcus. Very good. Streptococcus. You can say even the most easiest one, E. coli. Everyone knows E. coli. Who does not know E. coli here? Tell me. Tell me, then I'll tell you what I'll do with you. Everyone knows E. coli, right? So streptomyces, streptococcus, then E. coli, all of those are bacteria. Tell me the name of one virus.
Ma'am,
Coronavirus.
Coronavirus. Yeah, it is a virus. Very good. Okay, most of the viruses, their names are like that only. Like influenza virus, measles virus. So their names are like that. One name of fungi. Mariam, one fungi. You have studied biological classification from. Tell me. Any fungi. You people can't remember even one fungi. No one can. Okay, I'll give you one common name, then you people tell me the scientific name. Huh? That is good. Yeast. Tell me the scientific name of yeast. No one. The most famous yeast. Saccharomyces cerevisiae. Saccharomyces, right? That is a fungi. Okay. One protozoan. One protozoan. The silence, like I can hear silence. I kid you not, I can hear sun, sun, sun happening in my ears. It's like silence has a voice now. Tell me the
Ma'am,
name of one.
amoeba.
Yeah, amoeba. Very good. Okay, one helmet. Any worm. Name any worm.
Roundworm.
Hmm. Give me the scientific name of roundworm. You people remember Ascaris? Ascaris, the one that had a coiled tail and the one that had a long.
Yes, ma'am.
Okay, so I'm seeing your 11th. Not too impressed, students. Not too impressed. All right. Now, you people know what these are the major groups of pathogens that can infect us. Now, the pathogens, they enter our body by various means, right? They can enter by various means, and then once they enter our body, the first thing that they do before causing the disease, they start to multiply. So if one pathogen, like normally only one pathogen does not enter, like a group of pathogen comes, but they start to multiply. So from one, they become hundreds and then to thousands. And then when they multiply, then they start to interfere with the normal, vital, the important activities that we have. And pathogens, they have a property is like that they can adapt to the environment of the host's body. Now, if you people remember Animal Kingdom, which I know that you people don't, but still, I can assume that you people do remember. If you remember when we studied about, or when you have had studied about the worms, many a times it was mentioned that these worms have, they have this thing, hooks and suckers present in them. Why did they have it? So that if the worms, they usually attack the digestive system, the gut area, so they use it to clasp on our intestines or stomach bugs do that, so that they don't get washed away or they don't get moved away. So that is why they have that adaptation in their body, and they can adapt the environment of the host body. Doesn't matter whether it is acidic, basic, they don't have any problem with that. They can easily adapt. This much is clear, students?
Yes,
Okay, now
ma'am.
you people can note down till here, then we'll move forward. You can switch off your video and write down.
Ma'am, should we leave pages?
Yeah, you leave pages, pages, leave some pages, and then you start this chapter. Leave at least 3 pages for lac operon, or 2 pages, depending upon your handwriting.
Done, ma'am.
Done, ma'am.
All of you are done?
Done, ma'am.
All of you are done?
No, ma'am. 1 minute. Done.
Done, ma'am.
Ma'am, can you move up a little? Done.
Okay, all of you are done.
Yes, ma'am.
Okay, now moving forward. Wait. 2 more lines were there.
Done.
Done, ma'am.
All right. Okay. Now moving forward, let us talk about the bacterial diseases. Now, see, these are the bacterial diseases that you must remember. Like all of the examples that are here on your screen, these are to be remembered. Now, the name of the bacterial diseases: typhoid, pneumonia, then there is plague, tetanus, leprosy, cholera, diphtheria, dysentery, and tuberculosis. All of these are the names of the bacterial disease. Now, how do you remember these many names? So again, I have a mnemonic for your rescue. We have T, PPT, LCDT. T, PPT, LCDT. Just try to remember this. Okay. T, PPT, LCDT. Okay. So this T
Yes,
stands
ma'am.
for typhoid, P is for plague, then another P is for pneumonia, then this T is for what? This thing, tetanus, then this T, another T is for your, even I am forgetting. What is that? Tuberculosis. Yes, tuberculosis. L is for leprosy, C is for cholera, and D is for diphtheria and dysentery. So this is what you will remember, so that you can remember all of the names of this, like bacterial diseases. All of these are bacterial diseases. None of them are caused by any other pathogen other than bacteria. So cholera, again, from bacteria, leprosy from bacteria, tetanus from bacteria. So you must remember all of them. Okay. So this is the mnemonic TPPT-LCD. Now moving forward, we'll have to discuss each of these diseases one by one, but some of the important one only, I think so. Yeah. Okay. So when we talk about the bacterial diseases, we know that they are caused by certain bacteria. Now see, each of these diseases, they are caused by a different kind of bacteria. For example, if we talk about cholera. So this cholera, anyone knows which bacteria causes cholera?
No.
Vibrio cholerae. This causes cholera. If I say that the name is Vibrio cholera, then what do you think the shape of the bacteria would be?
Ma'am, comma like
Comma shaped.
Yeah, very good. Very good. Very happy. Yes, Vibrio cholera has a comma shape because Vibrium is the comma shaped bacteria. Okay. All right. So you people note this down. We'll not do a lot because you people still have to study about the chapter that is um, coming for your test. So I'll not do a lot in this particular chapter. So you note this down and then we'll see the rest.
Done, ma'am.
The
All of you are done, students?
Yes, ma'am.
Okay, do you people have any doubts up until now?
No, ma'am.
No, ma'am.
Okay. All right. So we'll keep it till here only for today. And in the next class, we'll do it with full speed. We'll do it with full interest and everything. Since now I just gave the intro and a little bit more, we have to go to the in-depth of this particular chapter. Okay. So if you people have any doubts, you can ask me in the next class also, and do well in your test. All of you, please attempt the test. Okay.